শিরূপা, হ্যালো শিরূপা,
বলুন স্যার।
বলছি তোমার ওই আইএসএম এর যারা পার্টিসিপেট করবে বলেছিল করেছে?
হ্যাঁ হ্যাঁ করেছে করেছে, আপনি নিশ্চিন্ত থাকুন, করবে। ইউটিউবে দেখবে, না হলে কিছু একটা হবে।
হ্যালো শিরূপা,
হ্যাঁ, কে কে বলছেন?
বলছি তোমার ওই আইএসএম এর যারা পার্টিসিপেট করবে বলেছিল করেছে?
হ্যাঁ হ্যাঁ করেছে করেছে, আপনি নিশ্চিন্ত থাকুন, করবে। ইউটিউবে দেখবে, না হলে কিছু একটা হবে। হ্যাঁ, কে কে বলছেন?
এই মনিকা হাসদাকে বলো তার এটা এটা মাইক্রোফোনটাকে অফ করতে। মনিকা হাসদা, প্লিজ সুইচ অফ ইওর মাইক্রোফোন। এই মনিকা হাসদাকে বলো তার এটা মাইক্রোফোনটাকে অফ করতে। মনিকা হাসদা, প্লিজ সুইচ অফ ইওর মাইক্রোফোন।
আমি যদি স্টার্ট করব কি আমরা এখন?
সবাই এসে গেছে?
101 জন জয়েন করেছে।
এক মিনিট পারমিতা দাঁড়াও। পারমিতা,
আচ্ছা,
একটু জাস্ট কটা বেজেছে? কটা? 3:05 হয়ে গেছে।
হয়ে গেছে।
ঠিক আছে। আমাদের মেয়েরা জয়েন করেনি?
মেয়েরা জয়েন করেছে, অনেকে জয়েন করেছে।
অনেকে জয়েন করেছে, টোটাল 101 জন জয়েন করেছে, 100 জন জয়েন করেছে এখন।
হ্যাঁ, আমার মনে হয় আরম্ভ করে দিলেই হয়, 3:06 হয়ে গেল।
ঠিক আছে, আরম্ভ করে দাও। ঠিক আছে, করে দাও, আরম্ভ করে দাও।
পারমিতা,
Good afternoon, everybody. I, Dr. Paramita Majumdar, welcome you all to today's national webinar on behalf of Department of Chemistry, Bethune College. This webinar, titled Today's Undergraduate Chemistry and Future Prospect, is organized by Department of Chemistry, Bethune College, in collaboration with IQAC, Bethune College. Now, I would like to request my dear colleague, Dr. Srirupa Banerjee, to kindly deliver the welcome address.
আনমিউট করো শিরূপা, আনমিউট করো নিজেকে।
Good afternoon to respected Principal Madam, Professor Krishna Roy, IQAC Convener, Dr. Kamal Kanti Shom, HOD Department of Chemistry, Dr. Gautam Bandyopadhyay, distinguished speakers, dear colleagues, and my beloved students. At first, I would like to thank our respected Principal Madam for her support and encouragement towards organizing this webinar. I, Dr. Srirupa Banerjee, on behalf of the organizing committee, would like to extend a very warm welcome to all of you to the webinar titled Today's UG Chemistry and Future Prospect, organized jointly by IQAC Cell and Department of Chemistry, Bethune College. It is my pleasure to welcome our distinguished speakers, Dr. Partha Charuti Dash, Associate Professor, IIT ISM Dhanbad, and Dr. Ashish Pal, Associate Professor, Scientist E, Institute of Nanoscience and Technology, Mohali. I would also like to extend a warm welcome to all of my dear colleagues from different colleges as well as of Bethune College. I am also very happy to welcome all of our beloved students, especially for whom this webinar is organized. In this never seen before scenario, which has been forced on us by COVID-19 pandemic, where all of us are physically isolated from each other, the webinar will provide us to interact with each other on the virtual platform. Particularly, it will provide us with the opportunity to interact with the students outside of the virtual classroom. In today's webinar, I am sure our speakers, with their wide-ranging experiences in chemistry research in both industry and academia, would enlighten our students on the importance of studying chemistry as a UG and PG subject with a view to further career options. We are eagerly waiting to hear from them because I am sure we will enjoy the next few hours with their lecture today. I hope today's discussion will inspire the students and certainly give some ideas about their bright future. Thank you all. Over to Paramita.
Thank you, Sripati. Now I request our head of the Department of Chemistry, Dr. Gautam Bandyopadhyay, to deliver the introductory. GB sir.
হ্যালো শোনা যাচ্ছে? হ্যালো,
হ্যাঁ স্যার শোনা যাচ্ছে, বলুন।
বলুন।
cordial welcome and heartful congratulations to all on the eve of 74th Independence Day. Good afternoon to our respected Principal Madam, Professor Krishna Rai, IQAC Convener, Dr. Kamal Kanti Som, esteemed speakers, dear colleagues, and my beloved students and staff. I, HOD Chemistry, Bethune College, would like to express my heartfelt thanks to our Principal Bethune College, whose continuous support encourage us a lot and inspire us to organize this webinar. Topic: today's undergraduate chemistry and future prospects. Similarly, I would like to thank our Secretary of Teachers Council, Dr. Amita Kaur, for her sincere effort, necessary help and cooperation to organize the webinar. I would like to thank the two esteemed speakers who have spent their valuable time for our webinar. And finally, we thank our students who are like salt and sugar in our activities. The objective of webinar is to introduce the students to some of the recent developments
lecturer.
in the field of chemistry that are important in the present and foreseeable future. The key objectives of the webinar are to reinforce in the students the importance and relevance of chemistry in the present day, especially in view of the current pandemic, where several challenges and opportunities that lie ahead for chemists in particular, to motivate the students to take up future challenge of the country in some of the frontier areas of the material science and medicine, to introduce the student some of the contemporary and emerging fields of research in our country and worldwide, to present before the students before the students some of the career options that are undergraduate training at our institute will prepare them through subsequent higher education in chemistry, will enable them to take up challenge, to make students familiar with the modern technologies in teaching and learning processes compatible to CBCS system, to encourage students in different innovative works in various fields of chemistry. I strongly believe that all the faculty members and the teaching staff of the college and students present in this virtual gathering will also enjoy the webinar. The webinar will involve two lectures by two important eminent speakers in the fields of chemistry who are very active researchers and whose contribution to the field of material science and medicinal chemistry are recognized throughout our country and abroad. Dr. Partho Sarathy Das will enlighten us with his medicinal chemistry of topic chemistry and life, a drug discovery perspective, and Dr. Ashish Pal will enrich us by delivering a lecture and insight into nano world and learning to mimic nature. At last, with these few words, I express my heartfelt gratitude and regards to all whose virtual presence, help, and cooperation will make our webinar to a great success. With this, I stop here and requested Dr. Paramita Majumdar to proceed onwards. Thank you.
Thank you, Gautam da. Now, this is the time for us to move on to the first lecture. The first lecture will be delivered by Dr. Partho Sarathy Das, Associate Professor, Department of Chemistry, IIT Dhanbad. To introduce Dr. Das, he received his PhD from CSIR National Chemical Laboratory in 1999 and did his postdoctoral studies at RWTH Aachen, Germany, then at Tohoku University, Japan, and Harvard University, USA. Dr. Das started his industrial career in 2003 with Dr. Reddy's Laboratories Limited, Hyderabad, and worked in Discovery Chemistry Group, having research focus on various therapeutic areas, example oncology, metabolic disorder, and antibacterial activity. Then he moved from industrial research work to academia and joined CSIR Indian Institute of Integrative Medicine. Jammu, where he served as Divisional Head of Natural Product Chemistry. At present, he is a faculty of IIT Dhanbad. His current research interests include medicinal chemistry, development of new synthetic tool, and synthesis of biologically active natural products. He is a recipient of Bronze Medal from CRSI in 2019, Pavlov Peer Review Award 2019, and Outstanding Reviewer Award 2019 from New Journal of Chemistry, along with many other awards. Today's topic for Dr. Das's lecture is Chemistry and Life, a Drug Discovery Perspective. Before I hand over to Dr. Das for the first lecture, let me request you all, please do disable your microphone and video for the smooth rise from Dr. Das's lecture. If you like in the question answer session, then please write them to please deliver his lecture.
Well, very good afternoon to all of you, and I hope I am audible. Yes?
Yes, sir, you're audible.
Yes, sir.
Okay, great. So I'll just share my presentation first. It is now visible? Is it visible?
Yes, sir, it is visible.
Okay, I will go into a full, I will go into a full mode. Okay, so yeah, so great to be in the afternoon with Bethune College, and thank you, Sirupa, for your kind invite. Action. Okay, so from historical figure like Kadambini Ganguly to Madam Chatterjee, the great organic chemist from India. Okay, so I feel honored to be speaking today in the webinar, and my topic is going to be on chemistry and life. And of course, I will take you to the journey of chemistry into the pharmaceutical world. And by end of the lecture, I think the student, if you feel it anywhere, some questions or anything, you can put it, I can answer after the lectures. You can reach me in my email, or you can WhatsApp me if you have any queries or any other perspective in this regards. Okay, so all right. All right, so I think as I told, that is going to be my topic on the chemistry and life, drug discovery perspectives. And so what I'm going to cover in this whole lectures, okay? So I think most of them, as Sirupa told me, most of them are undergraduate students. And so I have to make there some kind of career opportunities in these areas. So I will cover in such a way then you can have a fundamentals on this thing, chemistry and of course the pharmaceutical aspects. And then we'll discuss about the Indian context into it and where we can find the our future perspective in these aspects. Well, so if you think about the chemistry, for a student, it's central to modern science, almost all human endeavors, and then important to all science, and technology has been changed. So it's become completely interdisciplinary. So there is no chemistry, there is no physics, there is no mathematics, or there is no mechanical engineer or computer science. So everything is clubbing together, and the main purpose is the human well-being. So that's the overall purpose and overall goal. Of course, chemistry is a natural science, and that's the most interesting part of chemistry is, so it's a language by its own vocabulary. So whenever we say the chemistry is good, that means the bonding is happening. This is important with the molecules, as well as chemistry is important in the society for the bonding. Okay, and this is a way of thinking. So time to time, chemistry has been recognized by the worldwide. So 2011, we have celebrated the chemistry year, Madam Curie's 100 years of Nobel Prize winning. Last year, we have celebrated the international years of these things. I don't know how many of you can recognize this Google Doodle. So this has been 100 of for Madam Asima Chatterjee, which is the students of your Bethune, as I found it from your website. Okay, so I will encourage the student, please read about her. Okay, you will be encouraged. Madam Chatterjee, great organic chemist. The whole India actually learned the chemistry, the organic chemistry from her. Okay. All right, so let me start with a very fine picture. I will say it's a picture. So for the student, because this is a COVID-19 scenario everywhere. Okay. So if you find it, the student might have difficulties, but you can read it. Okay. Today, if you go to any places and ask anybody, do you understand the hydroxychloroquine? You will say, yes, hydroxychloroquine, understand. This is now people are using for COVID treatment. Or Fabiflu, like Favipiravir, which is in the right hand, on my right hand side, top of my right hand side structure. Or the very popular drug, remdesivir. Today also, you will find in the newspaper that Cadila has been marketed the remdesivir, the price has been reduced, or Pharmaceutical has been done with the favipiravir. Of course, we have one more name in the steroid molecules, which is dexamethasone. So why I showed you these 4 structures? Just to remind you this, where the context, where the chemistry is coming with the life. So this is indeed true, and this is extremely important. Where we have a chemistry, of course, there is a life, there is a molecule, there is a drugs, and there is a drug discovery. So this my whole lecture is going to base on the drug molecules and their importance, and of course the drug discovery, how it comes from the chemistry point of view. So if you ask any student, might be in the basic, when they are studying the BSc or come to the college, the teacher might have some time asked, okay? So, or the student can come to teacher, sir, madam, what is the drug molecules? So, of course, if you can have it from scientific language, the drug molecules is a pharmacological effect. So that's the word, pharmacological effect. So, and of course, it treats and cures a disease. Then only it's have a pharmacological effect. And definitely the drug molecules come through a regulation. If there is no safety parameters of a drug molecules, then there is a issues in the human body. So that should be regulated. So it has a pharmacological effect. It's come through a regulations. And to bring the molecule into a stage where it can be effective in the human body to cure a disease, the whole process is basically the drug discovery process. Now, to go into this picture, I would like to draw your attention and the students, particularly, or maybe the young faculty's members. So there is a difference between the generic versions and the patented version in the drugs. So generic drugs, what we are taking every day, day to day, most of them are low cost. So this is basically biosimilar kind of molecules. And India is the largest manufacturer of the generic medicines. Of course, we sell all over the world these medications. And these are the lead companies from India. You might have been taken many of these companies' molecules, how it has been effective in the human body. So why the generic medicine is important? Because it's a cost effective and it is affordable for the mass. So that's the things. Now, what's the difference between these two? Branded name and generic name. So brand name or the drugs must be patented. It should be exclusive right for a company. It's a new pharmaceutical ingredients, API, or it's a generics name has been started by the many companies into these things, or they can synthesize the molecules, sell at a cheaper price or out of patent. I just depict the two molecules, which is both are anti-diabetic. I think many of us familiar with metformin, which is an anti-diabetic first line of treatment for a diabetic, and linagliptin, which is under. So when the process begins and when the company brings a new molecules into the market, so that's become exclusive right for this company to sold it all over the world under the patent reasons. And when it goes out of patents, many company can come up with their own process and synthesize the dal, make it generics for the masses. Now, why we need a new drug molecules, or why we need a? So of course the questions can come here. This is the resistant could be one of the issue, because all of us knows that antibiotics become resistant, that oncology cancer drug became resistant, or maybe the new disease like COVID-19. So the whole world is searching for a molecules, for a drug. So less tolerance or the side effect, and safety and better efficacy. Safety is a parameters, and the whole parameters ends with human well-being. So focus on a new drug, basically with the chemist, which basically the first pillar into this success story of the drug discovery or the pharmaceutical world. So this is the safety versus efficacy we tell. So pharmaceutical products should be used when it is a benefit to a patient. Benefit takes into account both the drug ability to produce the desired effect, and of course it can keep aside the adverse safety effect. Now the question starts here. So where we the chemist is very important, and the chemistry learning is important. So I just put a cartoon diagram here for your understanding. Very, very important and very common for most of us. We take antibiotics. If you can see, the generations come one after one. The molecules come, like if you start from penicillin, the very old molecules. First generation, second generation, third generation, fourth generations. All molecules become efficacious and less side effects. And this is very useful for the human being. So now the question is that to come up with the new compounds, better safety profile and better efficacy, the process has to be optimized. And the chemistry and biology has to come together. Molecules has to be synthesized. And here is the learning process. Here is the chemistry play a very, very big role. Starting from the idea generations to molecule synthesis, to come up with the lead compounds, put into the animal model, come up with the efficacious compound, synthesize in a kilogram level, gram level to kilogram level, all process done by the chemistry people. And that is the beauty, and that is the learning of the chemistry, and that's why is the importance of chemistry today. It is so interdisciplinary today. The traditional way of learning the chemistry, whether you are studying the physical chemistry, inorganic chemistry, organic chemistry, or analytical chemistry, everything's clubbed together with the interdisciplinary areas today. And that is one of the big interdisciplinary area, the pharmaceutical science and as well as drug discovery. Well, so I always excite the students with their future perspective, with their careers. I just take the 4 compounds, the name, because nowadays neither the parents nor the students are excited about the subject, about the learning, if there is not a job opportunities. In IIT, students come in the very first day and next day morning start asking package kitna milega. So the same questions come for everywhere. Where is our career and where we should go? So you can imagine how big is the pharmaceutical world and how good and how wealthy is the. Where is the most question is coming, starting from cardiovascular drug. So which is one molecule is a $10 billion, you can convert into Indian rupees. Or the oncology drug, almost $10 billion. And then pregabalin, which is a neurological disorder. There are many molecules. Next slide, I will show you how the molecules, small molecules looks into the market. Sirupa, I can share with you this pictorial diagram. You can take a big picture of these things and put in your department, in front of your department. The student will be excited to see the molecular design, and as well as then they will see the tag of the how good into the market and where is the job prospect and how discovery and pharmaceutical companies are important for them to choose chemistry and pharmaceutical as a career options. Okay, so how the origin of drugs will come? You can find a student can today go and search, find in Google many of the things, like it start with the morphine and the first molecule urea started synthesizing, and then come quinine story, which is very common, +2 level. We always study the quinine. But I want to take you through a journey of salicylic acid. Very common molecules. If you start very first day, your BSc classes, the teachers tell about phenol, the teachers tells about Friedel-Crafts reactions, and then of course the teachers tell you Reimer-Tiemann reactions. Okay, from where you can get the salicylaldehyde from phenol, and then you can convert into a salicylic acid. A different way, okay, for a drug discovery or from a pharmaceutical world perspective. If I ask student today, maybe if I could have been present into the Bethune College, I can go into the board and then ask the student, oh, this is a salicylic acid. So you know the story of aspirin. If you can see the aspirin structure, it is basically the phenolic group has been converted into the acetyl group conversion. So acetyl salicylic acid, aspirin structure. Today, even today, this is 120 years old molecules. I am not reading the whole paradigm, whatever I have written here. It's most of them are available from the Google. You can go and search the history of aspirin story. But when it starts from the getting from the bark of a willow tree, then it comes to a salicylaldehyde and then convert into a salicylic acid. But the salicylic acids also have a properties. It can relatively help in the pain killing. But the problem when you start taking salicylic acid in your stomach, because it's a simple acid, you know, it will bother you, okay, in the stomach. So then chemists thought it, why can't we convert in such a way the buffering situations can come? Then the acetyl salicylic acids came. And the German chemist who really invented these things, he sold it to, he gave it to the buyer, the formulations, and it is first utilized in arthritic pain, you know. And then it becomes a wonder drug still 1889 patented, total 44,000 tons world over medication, world over requirements, wonder molecules. And the chemistry and the drug discovery starts with the very first salicylaldehyde to salicylic acid to aspirin. Of course, that we got from the nature, but this is the first molecule which has been synthesized by German chemist, Professor Ellis, the father of chemotherapy. Student can go and today search the magic bullets. They can find a lot of inspiration by reading about this story. And this is arsenic drug. It has some side effects, but this is the first synthesized molecules. White. First molecules, father of chemotherapy. Now, what is the difference today? What we exactly do today in the present scenario, when we think about the drug discovery, when we think about the medicinal chemistry, when you think about the pharmacokinetics, when you think about the molecular biology, when we think about the clinical trials, or when we'll think about the pharmacokinetics or other physioparameters of this thing. So it is a highly interdisciplinary. So what exactly earlier it happens? We have a natural product, then you isolate it, then you find a active compound in it like salicylic acid, then you convert it to aspirin, the story over, without knowing any biological target. So here is the question starts. So today chemistry is completely governed by biology. The chemistry has two areas of research today in the broad area. Whether as a chemist, you can compound, you can work on the pharmaceutical world, or you can help the materials world. Anything you do today related to this kind of two world. So the difference starts here. Synthesis of the compound, manipulation of the structures, you get a compound, put it. Suppose it has given for a arthritis pain. Without knowing what could be the biological target. So what is the difference today? So after the understanding of the molecular level of biology, the biology now we understand into the molecular level. Students can know ATP. Everybody start ATP, you know, what it play a role in a cell. I will tell you the story of ATP also. So when you understand the cell cycle, any cell division or any metabolic properties into the cell, so drug discovery starts from there. Understanding of the biology. So the disease, we should understand the disease rather than a treatment. Understand the disease rather than a treatment. There are many kind of pain into the body. Somebody hit you, you can get a pain. Somewhere you cut it, you can get a pain. You have migraine pain. You have some kind of other headache, or you have some internal damage, you can have a pain. So that same molecules cannot work for all these things. Same medication cannot work. So you need a different therapy. So this is, we now treat the disease with a biological target and possibly with the drug targets. I just want to draw your attention here. These are two molecules who have diabetic or hypertensive or some old at home, you know, very lipid lowering to cholesterol lowering medications. That is also one kind of treatment for the when you have a hypertensive. So doctor treated, you maybe have a blood cholesterol in the high level. So that is different. And of course you have to reduce your pressure. So you take a different medication. I just put MOA, mechanism of action of these two molecules. These are completely different. So here the modern drug discovery leads the story here. I just want to take the student into draw their attentions in these things. The molecules target in a different biology. So the mechanism is highly complex into the body. The molecule is going and hitting the target. That is the whole drug discovery phenomena. And today, when you think about the COVID virus, everybody is taking and coming into the lungs and binding with some kind of receptors. And then many phenomena are happening in the body. So if somebody want to block the receptor, then you can do some kind of purpose. So the same story also with some other therapeutic targets. So the biology and the chemistry has to combine together, and the marriage has to be very, very much harmonic. Okay. So then the drug discovery come and starts. Another things I think many of us take augmented, but none of us seen what the intermediate into it. It has basically two molecules combination, amoxicillin, each other in the mechanisms. So here we understand the molecule, and when the molecule goes into the biological target, then how the other molecules can help the molecules to find their biological target. And of course, in a certain amount, these molecules or drug molecules should present into a biological, it should show the efficacy. Can you imagine when you just think of a molecule, you are taking one drug molecules. So how many parameters, how many barrier it is going to reach to the target molecules? Your target is cell. It has to reach to the cell site or target site. Even just very blind way, if you can think of that, if you take a molecule today, any drug molecule, simple crocin, paracetamol, you check into the stomach, stomach pH is 2. So it is tolerating in the stomach. And then it is entering through intestine into the body or into the blood circulation, and then can showing the effect of these things. So we'll discuss these parameters. So this is a learning for the student. So it can come with a the whole is basically huge teamwork. This is when you think about the drug discovery, the pharmaceutical world, the pharmaceutical companies, we think how this thing work out basically in a pharmaceutical setup. So when you form a research team, you can come up with a molecules synthesized by the chemist. You can go for the testing of the molecules with the animal model. Then you do the formulation study. So compounds, molecules can come to you in a tablet format, gel format, injection format, solution format. So here we do all this story. Then the most interesting part, student can pay attention. In India, we are very good in formulations, very good. Actually, we are kind of things where we can sell the formulated product to all over the world. When everybody was struggling for hydroxychloroquine, India, I think, supplied to 150 countries. Can you imagine that? With a short span of time. We are very good. And then we are very good. Why I am telling we are very good? Because there are a lot of job opportunity. As I told you, so when you start reading, when you come to the, you see the job market, where exactly and where the, so today thinking is exactly your chemistry has to lie where the job market had more openings. You should focus in that area. Because this I learned from my industrial career. I started in industry. Almost 10 years I worked in a pharmaceutical industry. So then the job opportunity comes here, and another job opportunity comes when these regulation markets start coming. I will show you a few slides in the regulations. And particularly this is very good for the girl child, where the patenting of the molecules, the regulations happens, and in India, after the patent regimes, we are doing a very good in this area, and pharmaceutical company actually look for young and bright people in these areas. So if you have a background on chemistry, if you study the law on patent, you have a huge opportunity. Or if you can study the regulatory affairs, it is not like every time you have to study organic chemistry or physical chemistry, you have to come to IIT or you have to go to IAC cultivation of science to do the PhD and do the things. But I'm telling these are also the opportunities. And girl child do much better in these areas. And today if you go to any pharma company, I will ask the student to scroll them to see the pharma company and see who are leading this kind of things, either the patent or regulatory affairs. All ladies. They are highly qualified, highly competent. Okay, so once it has done the molecule, so we can go for the clinical trials. Nothing to be asked for the scientist here. All the doctors do these things, the clinical trials. So phase 1, phase 2, phase 3, and then the molecule launch into the market. That's why today you are finding we are always saying when the COVID things will come. I'm not talking about the vaccine today here. I'm talking about the small molecules. But even vaccine also need this kind of clinical trials. That's why the process is so complicated and so time consuming. These are the molecules approved by FDA, which is Food and Drug Administration, which is a regulatory body, the largest market of the world, the US market. You can see the number, and from time to time it varies and depends on their market potential and their other potential in a therapeutic. This is a drug discovery environment, how really the times, it is almost 10 to 12 years, the development process. Success is very less in these things, but cost is also very high, but also the reward is very high. If you have one molecule's $10 billion product, you can imagine it. You can calculate the cost, which one you have, and what is your profit into these things. Here is I would like to take you to the in between chemistry and biology interface and other areas. So where we have done the organic chemistry, and then we synthesize the compounds, we make the molecules, we do the medicinal chemistry with the help of biologists and pharmacologists and other pharmacokinetics people. So then we come up with the compounds, which is called heat to lead molecules. or it can, with a desired pharmacological effects, with a efficacy. So that's the overall story here. And this is a iterative process. As long as you are not coming up with the right molecules, you work on the pharmacology, you work on the pharmaceutical profiling of the molecules, and vitro or in vitro, which is a enzyme and the receptor. In vitro is basically open enzyme, and you can do inside the glass, and in vivo is inside the animal. So once you have a good molecules from in vitro, we go for the in vivo testing with a better pharmacology and better pharmaceutical profilings. So these two, this is a average from your start from bench to the market, and you can see the amount of molecules or amount when we synthesize the compound, starting from gram levels to kilogram levels, even ton levels, this compound, and over 12 to 15 years history for a new drug compound. This is a number of compounds we synthesized into this thing. So organic chemist has a very, very good opportunity in pharmaceutical companies. Extremely important in India. We have a lot of companies and huge job opportunities. Okay. So we start from basic research, we go to preclinical testing. Okay. And then we come up in preclinical testing, we have, you can see the number 3,000 to 10,000 molecules for a target, biological target we synthesized. And then we go for the preclinical, and eventually we go to the clinical trials, 1, 2, 3, and then market, we marketed only one molecule. How the concepts start from bench to market. So it has two aspects. One is a discovery phase, which is, we called it preclinical phase. And another is the development phase, which is a clinical phase. Maybe it is difficult for the student to remember. So please remember, there is in a two phase. One is preclinical, another is clinical. One is discovery, another is development phase. And the modern drug discovery, basically it's a team effort. So you can see the team players are 12 team players, like it's a total cricket team or football team. It starts with the medicinal chemists, it goes with the molecular biology. So when you enter this kind of arena, or as a professional, or you want to work in this area or pursue your career, you really love to work it, you know. You really, really love. So how many professionals you will interact in day-to-day basis? If you are, suppose if you are a medicinal chemist, you can interact with molecular biologists, you can interact with the molecular modeling guys, with the computer where the designing is happening of the molecule. You have intellectual property, as I discussed. We'll discuss about the pharmacokinetics and drug metabolisms. You can have a formulation people who basically formulate your molecules, in vivo pharmacology, toxicology, regulatory affairs, or process chemistry and engineering, where your molecules are going to into the plant. And of course, you have a clinical data when you have a clinical trials and data management, and you can also mix with the marketing people. So huge learning process in these things. You will love to work in these things as a team members. Of course, the chemistry leads the, so that's the in pharmaceutical world, it's a medicinal chemistry. So where you have a synthetic chemistry, pharmaceutical science, process chemistry, of course, drug metabolism, physical chemistry, who do the physical other physio parameters they observe, or IP and legal, all these things are associated with the medicinal chemist and the chemistry. So drug discovery, basically we the medicinal chemist actually work on the two areas. So a student can just for their curiosity can remember these two words. This is pharmacokinetics, which is we basically observe here pharmacokinetics and pharmacodynamics. So pharmacokinetics is basically when you put a molecule into a drug into a body, so what the body does to the drug, okay? Or pharmacodynamics is basically what the drug is doing to the body, you know. So that's the balance you can understand first. So the pharmacokinetics and pharmacodynamics. Okay. So once we have a ADME, absorption, distribution, metabolism, and excretion, that is the 4 properties. And another is the pharmacodynamics, where we have a plasma concentration, target the molecules into the right direction and right therapeutic target. Okay, so these are all bookish knowledge. So you can find it when we study the medicinal chemistry. Yes, so actually, when I start giving this kind of popular kind of lectures, and very, I think last month I talked the people in Krishnanagar Government College there. So then actually I requested the faculty members and of course the higher authority of the college to include at least 1 chapter in their chemistry, in a medicinal chemistry, which is very, very relevant today. And maybe the, I repeat, the medicinal chemistry for the student, 1 chapters. I hope the higher authority is listening to me, and with the perspective to industry, or yeah. So the sorry is we have some issues. Okay, so once we have a this learning with the perspective of industry, so then the students will be motivated, and at least they can find their career path or career opportunity. They can learn. So yeah, today many industry peoples are very good, and they can come even to, I told already to Sirupa, so even I can arrange for you, and they can come and they will be happy to, because you are locating in a prime location in Calcutta. So they can come and they can give a popular lectures, and where student will be motivated to find their career opportunities in pharmaceutical companies. Okay, so the issue and challenges with drug discovery remains when we go for a drug likeness of the molecules. The molecule has to be drug-like. We follow the Lipinski rule. I'm not going into the details. Students have more interest. You please type in Google and find Lipinski rule in the medicinal chemistry. You will really love it. You know, it's called rule of 5. Okay. So no details, because otherwise I have to show you many other things. Okay. So, so this is a little bit from the industry perspectives. So how the industry works, and deciding on an area of research, it should have a commercial aspect, because if it doesn't have a commercial aspect, the company and the benefits will be collapsed. Okay. So you have to choose the area of research where is the important. Today, everybody wants to work for the COVID vaccines. Why? Because you know its demands. Okay, so or COVID, like where the situations is you can improve the better and better molecules in the drug properties for the COVID treatment. So that's the deciding on the area of research. That is very important for a pharmaceutical company, when you will be where it is important for the time when the disease is important. So of course, this is an unmet medical need and commercial opportunity. So both these things has to go time to time. So this preclinical stage, it is the flow. It was works, the genetic biological targets. Then we go for hit to lead molecules. Then we go for pharmaceutical properties. Then we go for the lead optimization candidate and the clinical trials. This is the medicinal chemistry. It works with the receptor and drug design. So when it has a drug design and receptor molecules, it's just like a lock and key process. So you have an enzyme. In next slide, I will show you how really it works in a oncology drug. Where you have an enzyme, the molecules is going and fitting into the enzyme pocket. Either it is a inhibitors or it is a acceptors. So that's we called it pharmacophore. So medicinal chemistry works in two ways. It can be conventional or it can be rational. So like random synthesis, or it can be structure guided drug design approach. So all these things comes to find a low nanomolar compound. And nowadays computer science, structure guided drug design, artificial intelligence, machine learning, all these things are extremely important for drug discovery process to come up with the quick and hit molecules, quick and fast. So that's the things, how quick you can come the molecules, and structure guided drug design actually help in these things. So so this is a huge area. The physical people, physical chemistry people who have interest, they can also find a lot of opportunity in these things. Okay, and then we go for the biological assay, where many chemists converted into the biology. You know, most of the good biologists today all over the world are chemists. Most of the biologists, because they understand the molecular aspects of biology. So we do the in vitro, in vivo, ex vivo testing. This is the animal models have or disease models. Okay. So this is typical biological assay. I'm not going into the details of this assay, how the assay is performed. And if I will request the school committees, so if you really want to learn the biological, student want to learn on the biology aspects of the assay, or particularly who are studying the life sciences, you can invite some people who actually work closely with the industrial setup for the biological assay, so they can give you better picture of all these things. Suppose if you have a target called kinase, I just want to take 2-3 slides. In kinase is a very good target for oncology. You know what it exactly happens in biology. It's, you can remember ATP. Actually, ATP is the energy which transfer the phosphate group to the enzyme, and the cell division starts. So if in oncology setup or in cancer setup, what happens if the molecules irregulated cell growth happens? That means you cannot regulate the cell growth. So the kinase is highly active in this thing. So you have to block the kinase, this ATP, this phosphate group transformations, or this thing. So how to block the things? So this is a story of Gleevec. This is a story of Gleevec, myeloid leukemia, blood cancer. The historic molecules, the first kinase inhibitors, it's called Gleevec or imatinib from Novartis. So this is the active protein is BCR-able kinase. So ATP comes and bind into the pocket and transfer the phosphate group. Leukemia happens. But if you have a molecules, small molecules, which is basically mimicking the ATP part, it comes and block the space. It can out the ATP from there. No signal. Cell division doesn't happen. It's a cell death. So no, it's a blockbuster molecule, saves millions of lives. Please study the story of Gleevec or imatinib. This is a imatinib structures, how it's really binds into the pockets, how really do in the molecular biology. So this is the kinase, its treatments for the myeloid leukemia. This is a gefitinib, which is a molecule target for the lung cancer of these molecules, and molecular biology and all this study, you can find it. Today, if anybody has interest, study on the kinase inhibitors, more into these things. So we basically the medicinal chemists do the structure activity relationship study of all these molecules in this fashion. So here is the organic chemist is extremely important. If you have expertise in organic chemistry, you can do this kind of structure activity relationship or synthesize these molecules. These are the marketed kinase inhibitors. A lot of molecules has been marketed. At least 50 compounds has been so far as a marketed drugs. Well, drug metabolism is important, because why I am showing all these parameters. So these are every department in a drug discovery setup. So So these are the all future opportunities for the young minds or the students who are studying particularly today in the graduate levels. So that's really the future perspective it would be. So drug metabolism actually works on this. How the story is that almost all drugs actually go metabolized by liver. Okay. So we have to understand the metabolism part of this thing. So if the metabolize of the drug very fast, quickly, so it is very difficult to go into the blood circulations. So we have to stop the metabolism. So then chemist, the metabolic scientist, we do this job. So this is a good opportunity for the student to learn this kind of techniques, how this enzymes and how this part works in a drug discovery setups. This is a pharmacokinetics as we understand the pharmacoadsorption, distribution, metabolism, and excretions. Good job opportunity. And NIPER, there is a organization called NIPER. There are 7 NIPERs in all over the all India, and we have a very good NIPER at Mohali. So they have a lot of this kind of courses they offer, particularly in pharmacokinetics and drug metabolisms. So if you have even after BSc, you have to check it whether how you can enter into this kind of colleges or to learn these techniques and get their degree and for a better opportunity in pharmaceutical companies. This is a pharmacokinetics parameters, typical parameters, how we really quantify the units of the drug molecules in a plasma level or in a plasma concentration when it has to reach to the tissues, like peak concentration, T-half values, all very, very technical terms. So interested student, please study the pharmacokinetics if you have more interests. And of course, the pharmaceutical profiling of the molecules depends on the so many techniques and so many technological terms. Why I bring this kind of technological terms to you? Because you have to be expertise in this area to have your job opportunities. Okay, and to learn the techniques, you have to understand the science and engineering things. So you have to go to a good school or particularly where this kind of course or this kind of teachings or this kind of research happens. So that will make you perfect. So today, as we tell always, today the student should not be degree ready. You should be industry ready. Okay. I repeat, you should not be degree ready. You should be professional ready or industry ready. So then you have the opportunities. Well, so these are the lead optimizations when all technical things, when we have the heat molecules, to go for the lead molecules and drug-like compounds with the structure activity relationship study. Safety parameter is extremely important. Okay. And I don't want to discuss in details. This is, I think many of you know who are the senior colleagues to us. So this is a story of thalidomide story. If it is, these are the baby born with a fetus defect. If the drug molecules are not gone for the toxicology data properly, that's why this vaccine is taking time. It has to come with a proper shape, with the after proper thorough checkup in the safety records. So safety is extremely important. And that's why drug is basically the definition is a drug of a safety versus efficacy. So safety is important parameter. So we understand the safety parameters. It goes the various toxicology tests like 14 days, 1 month, 3 months, or even better if it is required for a molecules. First it goes for the animal model, then of course it's enter for the human body. Okay, these are the therapeutic index we basically do to understand the safety profiling of the molecules. This is a regulatory term. So when it comes the molecules to the lead and lead optimization, so it's go for the regulatory, and we have a good GLP, which is molecules come for a manufacturing process. This is called GLP, good manufacturing practice. And here the chemical engineering and chemistry actually combined together, or even the mechanical engineering people can play a big role in the GLP manufacturing process. And this is a clinical trials, which is phase 1, phase 2, phase 3. This is nothing, all the doctors do all these stories for us. Phase 1 is a healthy volunteers, phase 2 is a safety and efficacy with a minimum, phase 3 is the largest. That's why you find you might be, all of us study when the molecule of AstraZeneca in the vaccine, it goes from 10 safety profile to 100 for the patient and 1000 for the patient. So like this. So the whole thing works. Okay, once it come out from the phase 3, it can be marketed. Okay, so this is the overall process of drug discovery. So you have a pre-discovery, you have a drug discovery, then you have a development process, clinical trials, FDA approval, scale up, and manufacturing, and then it comes to the market. Overall, 10 to 15 years story. Okay, and the team game. So what exactly in the whole process when you work for a industry like of setup, it has a go and no go decisions. So this is also good learning for when you work for a team, where to stop, where to go, where to no go, where to take the what kind of measures you take. This will make you perfect professionals to have a good career opportunity with other industry also. This is a success story, how a typical company works and typical good compounds. So it's a good drugs, effective development team, good drug development. That's the overall story between the pharmaceutical industry. This is an Indian perspective. So we started in stage 1, early manufacturing process, 80s, and eventually we come with a development process on 90s, and 2000 onwards we are really in growth phase. The pharma companies are in growth phase. We are now in stage phase. Up to 2002 onwards we are very good. We are very good actually. We are doing the supplying the molecules to all over the world. Okay, and these traditions will keep on going on. So this is a Indian pharmaceutical markets, which is very good. I think it's almost $55 billion market today, if you can see 2020. And this is a very growth phase. We have, so why this is important? Why I'm showing you? Job opportunity. Job opportunity. You have to think the career prospect. Okay. Otherwise, today, chemistry is important to you. You are learning. Tomorrow, you will find, oh, chemistry is dull to you. Okay. You may not find that much interesting. So this kind of, you have to tag everything today with the market, how the market is doing or where the opportunities are coming. Okay, so we have a good contract research organization. So many people, I think, who has done PhDs or they search for a job, sometimes they are very good companies. They work for other pharmaceutical companies and create a lot of job opportunity for the students. This is an Indian advantage. After US, we have the largest number of manufacturing plant in India. That is the things, and that's why the biggest job after IT sector, we have created a lot of job opportunity in pharmaceutical sectors, and that's indeed true. So this is a global challenges and India opportunity. Always we say it's a cost cutting and a lot of young minds and brilliant people, academy, industry interactions. So of course, the Indian pharmaceutical industry has a potential to bring affordable and accessible drug for the masses. That is indeed true. I think many of us doesn't know. Today, the half of the African population won't be there if Indian company doesn't supply the HIV, the AIDS medication to with a almost free of cost, because neither the big companies from the United States is going to give you the drug molecules with below their sell price. We Indian company lot of do this kind of generous activity. Okay. Well, this is a good for the student. This is a good slide for the student where you can find your fellowships, because to study higher study, you need a fellowship and you have to enter. And these are all the, actually, when I was talking to Sirupa told that I can show some kind of data where we have the research institute, And then you can have a lot of sites where you can enter into the site. You can find out what is the criteria and what kind of qualifications, and when they have do this kind of all India level examinations, where you can find your opportunity to enter into this leading institute in India for your better academic records. Well, this is, I think I cover last also in this college, because this is an opportunity for the young researcher or young faculty members who are interested to pursue their research career, because this is an appeal to the opportunity to the faculty, young faculty members who have a very good track record of the research. They can come and do their research with the IITs or ICERs, which is Teachers Associate for Research Excellence, called TERE, and funded by completely DST and SARP. This is 3 slides I have remaining now. So this is a regulatory affairs. I will ask the young student to, if you have a pen and white paper today with you, just write down the words regulatory affairs. You might be forget. So regulatory affairs, then you can find it. So more about these things, how really it helps in the career aspects. Or there are various regulatory bodies which can really helps you to understand the regulatory world and the job opportunity and the career opportunity in these things. This is a harmonizations where the GMP and all these things work, and you know how they regulate the whole process all over the world. You must study of all these things. These are maybe very, very new to you in your study. Okay. This is a patent and World Intellectual Property Organizations, how the patent law and all these things, and where in India you can get a diploma on patent. This is basically offered by the law college, not the scientific institute. This is the law college they offered. And there is a Hyderabad, there is called Nalsar, and there is one college in Nagpur, which are very famous. Top college in India. You can look into these things. Even after your BSc, you can study this kind of opportunities for the girl child. Okay. All right. So I think I almost end of my story. So this is all human well-being, chemistry, biology, everywhere, whatever you do and whatever so far I speak to you, everything, all the target, the human well-being, all these things. And every lectures I would start it with the drug discovery process, and I would like to show one slide which I always love to share with you. This is the 3 very interesting pictures, okay, of the 3 history. Okay, so of course the first picture all of us know, the father of Indian chemistry, and but I would like to tell one words about him. Okay. Sir P.C. Ray is not only extra father of a chemistry of India. You can see he's a great entrepreneur. Okay, if you read about him, you can find a completely new about him. Today, you read an angle of his entrepreneurial skill. Today, Bengal Chemicals, Bengal Pottery, Calcutta shop. Today, the India is learning the Atmanirbhar Bharat. So he could give a directions to you even 120, 30 years back. Can you imagine? Great entrepreneurship skills. Sir P.C. Ray. Okay, so drug discovery we started. Actually, India started the drug discovery being 100 years ago with urea stibamine, Sir UN Brahmachari, Kalajar. You can highly inspirational. The doctors who lives in Kalajar. Another story. So this is a might not be knowing about his name, but the people who are from Calcutta University, they can take pride. Today, the rehydration therapy, if you have a stomach upset, the formulations what you approved by WHO, World Health Organization today, Actually, this is credit goes to Professor Hemendranath Chatterjee. You just see the paper, the scientific article which he published in Lancet, 1953. 1953. So I would like to conclude my lectures with these three inspirational stories, and starting from chemistry, learning of the chemistry with a drug discovery perspective, and eventually it can lead to the human well-being. I hope students will be motivated to find their future perspective and future course by learning chemistry in interface of human well-being. Thank you all. Thank you for the opportunity, and thank you listening to me. Thank you very much.
Thank you, sir. Thank you very much, because this was such an inspiring lecture for all the students. They can get motivated by listening to this, and actually all of us can get motivated, know many things about drug discovery and this academia industry interface. Okay, there is one question probably. No, there are no questions. There are no questions from the students till now. But if you still have anything to ask sir about this lecture, you can always ask him through email. Sir has already said this. Thank you, sir. If they have any queries, I think they'll put them, write them in their email or through WhatsApp probably. Thank you very much, sir.
Okay, thank you for the opportunity, and I hope I exceed my time. I'm sorry for that, but yeah. Thank you all for the opportunity, and I am now stop my sharing. Okay.
Okay, so then we'll move on to the next lecture. The second lecture will be given by Dr. Ashish Pal. He is also an associate professor, Institute of Nanoscience and Technology, Mohali, Punjab. Dr. Pal has completed his BSc from Ramakrishna Mission, Narendrapur. He has done his MS in Chemical Sciences from IISc, Bangalore. He has done his PhD in Organic Chemistry also from IISc, Bangalore. Sir has done his postdoc, two postdocs, one from Eindhoven University of Technology and University of Groningen, both from Netherlands. He has received a number of awards, prizes, certificates, and fellowships like SERP ECR from DST SERP, UGC DA ECSR from BRNS Young Scientist Award, and he has been regularly publishing his research articles in internationally renowned journals. The title of today's lecture will be An Insight into Nanoworld and Learning to Mimic Nature. I would request Dr. Pal to please start his lecture.
So, thank you, Paramita, for nice introduction. And let me share. Okay. So I hope the slides are visible now.
Visible,
Yes, sir.
Okay. So at the outset, I would like to thank the principal, Dr. Roy, and also the HOD of Department of Chemistry, Dr. Bandyopadhyay, for extending this invitation to be a part of this wonderful webinar especially for the Bethune undergraduate students. And also, I would like to extend my special thanks to Dr. Shirupa Banerjee, who coordinated the entire invitation and also other details. So, and I think I thoroughly enjoyed the previous talk by Dr. Das, who actually took us through very interesting journey of the drug discovery and the implications and also the opportunities which are which are present for the students in future. Of course, being when we were also being a chemistry student, we actually did not know where to go. We always thought it's actually the IIT or doing some research. We, frankly speaking, did not have much idea that, we can actually, we knew that, yes, we can go into the pharma industry, but, pharma industry has so many different aspects and so much opportunity. That's something I think many of us from Bengal, we should now cash on, particularly when we see that there are these pharma industries in north, let's say in Chandigarh or Hyderabad or Pune, they are thriving. So we should also have something like that. So with that, so when I was learning his talk, I also changed my talk a little bit because he showed very interesting scholarship opportunity, which I also had. So I just changed it at the moment because otherwise there will be duplication. So what I did is that, so in my talk, I'm going to talk to you first 30 minutes, something very general. And as he said, rightly pointed out, the pharma industry in India is really doing well. And that we all know that this has been and another, area where India is making its name after the IT industry. And there's also another booming world, that is the world of nano, which has also, taken its shape in last decade. And so that's what I'm going to discuss to you about how much opportunity the nanoscience can endow we with. And then towards the end, 10 minutes, I will discuss about how we are contributing into these nano sciences. Okay, to begin with, I think our institute is relatively new. It's just 7-year-old institute. So this is our temporary campus, which is very close to, if you know, the PCS stadium where all the cricket matches happens. It's hardly 200 meters away from there. That stadium. This is the proposed new campus, which is coming up. Actually, it has come up, and this is the one which is taken just a week ago, and we are currently shifting into this new campus. And in fact, we are going to have our Independence Day celebration tomorrow in this new campus because we are now in the process of shifting. So I just wanted to give you a flavor. Like, I think being a chemistry student, you are always used to seeing typical chemistry lab with burette pipette, filtration setup. But the modern chemistry, it is not only limited into that. It is also much on instrument based. And many of the things, if you see, this is a device where you actually do your chemical reaction within a flow setup. So that is something very interesting that I will come to you when I discuss something called kinetic equilibrium, kinetically controlled, as you know, thermodynamically controlled product, kinetically controlled product. This is useful to generate some of the kinetically controlled flow setup based reaction product. Similarly, you can also see here, I think this is earlier days, people used to, if you look at in this kind of dresses, you would probably think that it's like a space suit. But nowadays, because of this COVID-19, this kind of, you get to see such kind of dresses, and you already know that these are all actually PPE kit. So yes, this is a typical clean room or fabrication lab where you don't want any of the dust particle to be in, because you are actually making some devices. So what we do in chemistry, we do some reaction. That reaction we can do in controlled way in a special setup here, as you see. And also after doing the reaction, whatever chemical product you make it, if you can make it useful, you can also use it for different applications. So one such thing is this device application. You can actually make your chemical product onto some certain device for different various applications. This is just to give you a feel of how it looks. So a number of interesting personalities visit to our new institute that is dedicated for nanoscience and technology research. You can see some of these Nobel laureates over here. I think you would all recognize them very well. And that's my small group which I have developed for last 5 years or so. This is just to give an introduction about our institute. And apart from doing science, we are very actively pursuing this outreach lecture or the popular science lecture, which I'm actually giving you here. And when I visit my home in that is in North Bengal, I keep visiting many of the schools or colleges, particularly remote area, and also try to inspire them through some of the popular lecture. And we also do take them to, this you can see that they also visited to the, Birla Planetarium, so that from the remote area, we can actually pick up students and we can make them motivated towards science and technology. So with this, now I have a question to ask you all. So what do you want to be in future? So, well, definitely you are in, you are actually doing your honors in chemistry with some subjects as physics or mathematics. So definitely you must be one of the option is to carry research, to pursue your career in research and development. That means being a scientist, and apart from the typical academic setup. So I just would like to share with you what are the qualities you require in order to be a scientist. Basically, you need to have a very good intuition. You need to have, second important thing is observation. And third important thing is analytical power. Now you may ask me why. So when you do, let's say talk about chemistry aspect. So you have particular product reactants, let's say A, and another reagent, let's say B. You mix A plus B and it forms C, right? But now there are 10 different ways of doing this particular experiment. Now, if you're a good scientist, you would always logically think, and your intuition will actually guide you, that out of the 10 different ways, which are the first 3 possible ways you must try. Okay, so that's a logical connection you make and you try that out. Because if you try to study all 10 different way to do it, you really need a lot of time. But as you know, in living world, we are all limited by time because we have a finite age limit. So we must finish. So this is very important. Intuition is very important. Now, next thing is that when you do an experiment, observation is one of the very important skill, and you will see that 2 of you sitting next to each other would have completely different observation, and that's what makes discovery or science quite different, because someone can actually look at it very objectively, or that means without any bias. But the other one cannot really look at it that way. Second thing, third important thing is that whatever observation you make, that you need to analyze. That means it comes to this data interpretation. So these are the quality, if you have, you can actually plan an experiment, execute it, and then interpret the result, and you can actually publish it. So that is what actually a scientist needs. Now, if you want to be really good scientist, one of the things very important is that you need to have something, a creativity. That is what we think that person who's studying with arts, fine arts, they would have. But people who are studying science, that means they should have very good reasoning or analysis. But if you club them too, if you have creativity as your soft skill in your life, and you are actually studying science, then you actually balance your right brain and left brain, and you can actually create very interesting stories, and you can think something out of the box, and that is what is required for Indian science to progress. And this is one such story. I think I must tell you, everybody knew that blue sky, but the blue sky become red, particularly during sunset or sun rise time. Why is it so? And then, exactly 100 years ago, it was more than 100 years ago, Lord Rayleigh actually described it, and all of you know it from your class 8 study of the diffraction of the light by the dust particle. Different wavelength actually reaches to your eyes at different time, and that is how you can actually get this. And later on, as you know, staying from Calcutta, and Sir C.V. Raman also has discovered this Raman effect, which is also related to the scattering of light from ICS Kolkata. So now coming to the topic. So as we saw from the previous talk, yes, pharma is also very important. And another important aspect is nanotechnology. And as you see, for last 50 years, nanotechnology is growing, and it is growing something like, you can see the sigmoidal pattern, which is something like an exponential growth. I think this exponential term nowadays in COVID-19 era, many of you know it very well, this COVID-19 and the infection, it is growing, exponentially. There is something called doubling rate. I hope these are terms which are very common to you nowadays. So you see, this is really the research in nanoscience and technology is really picking up a lot since 2000, and it is expected that next decade and all the nanotechnology based products are going to be in market in a big way. Already they are in market and they are going to come in big way. So before going there, we must know what is nano. Everybody talks about nano. So when we talk about nano, that means we are talking about a particular dimension, okay? Or it can be also the time scale, but let's talk about the dimension first. So what is nanometer? It is 10 to the power minus 9 meter. Now, just to give you an idea in that perspective, so if you can consider our Earth and your football, and you see it is actually 1 crore, 10 million times smaller, or 1 crore, I guess. But if you take a football and then you consider the first, one of the first discovered nanomaterial, that is discovered nanomaterial, that is called fullerene, C60. I think all of you are used to this fullerene. So this is like 100 times smaller than the football. So that's a perspective I can give. And fullerene is actually in this something like 0.7 nanometers. So it is in the nanometer regime. So typically, even though people talk about 200-300 nanometer particle or dimension, also nano regime, but typically for typical nanoscientist, classical nanoscientist, this is the dimension we always care about, that is 1 to 100 nanometer. Now, even though fullerene, as I was saying, this is one of the first discovered and characterized nanomaterial, later we came to know that yes, nanomaterials were already there with us from the history. And if you ever go to British Museum, you can actually see this kind of Lycurgus cup, which you see it is, which can also change its color from a different angle. You can see in many of the Western church, you can see there is a particular glass windows. Here you can see there are different colors. And in fact, these are all actually the gold nanoparticles which are impregnated in the stained glass windows. And so this was there from a long time ago, but just because we could not characterize them, we just said that these are probably some colloidal dispersion. So as we all know what is colloid, I think you were studying in your undergraduate chemistry, but yeah, so can I take this question now, or there are some? So I guess I will discuss after the talk is over. That's because some people have raised hand. So let's discuss after the talk gets over. Now, one of the things you can see, these are this colorful material. These are all gold nanoparticle. So these are all gold. Okay, yes, please go ahead, ask. I think.
Sir, please go on.
Okay, fine.
Question session will be after your talk.
Okay, that's fine. Thank you. So as I was discussing, these are gold nanoparticle. Very interestingly, if you look at your bulk gold, all of you, I think if you go to jewelry and all, shop and all, you actually get to, you're familiar with this gold, which is in bulk. But this particular bulk gold, you can actually break it down and you can make it in the very small dimension, that is in nano dimension. You can see this color, the physical property completely changes. Why? Because the surface to volume ratio in the bulk gold, that completely changes when you actually go to this nano regime. And another interesting part is that as you can change this size and the shape of the nanomaterials, you can also change or tune the color of the gold nanoparticle. So this is this now, being chemist, I could not resist myself. I thought that, okay, since I'm discussing, gold nanoparticles, it's very easy to synthesize gold nanoparticle. So what you need to have is that you need to have a gold salt, that is HuCl4Cl4. And being a chemistry student, you know that here Au is in +3 oxidation state. What you need is that you need a citrate, which is a reducing agent, and which will also cap the colloidal growth of the nanoparticle. So this is what happens. So it actually reduces the Au3+ into Au0, and then as soon as it goes into Au0 metal, it will actually start growing from all direction. Then that's how this charged up citrate ligands will actually cap this at this particular pH, and it will actually stabilize the particle. And you can actually, this is the thing, you can see that this is initially, this is the Au3+. As you add this reducer citrate, you get to see that there will be a nucleation, and then after that there will be growth with time. So you get to have such gold nanoparticles, which actually you can see only by this transmission electron microscopy. So this is how you actually characterize the nanomaterial. So you need to have an electron microscope over here just to visualize such nanoparticle. I'm going to come there when I discuss something about this nano world. So this is just the introduction of the nanoscience I'm going to give you. Now this is another important slide where you can see very interesting slide where you can see that how nanotechnology has revolutionized the world around us. And you can see in information technology, like I think many of you have seen the TV earlier days, maybe 20 years ago, or you might have seen the laptop or the desktop computer. Nowadays, everything has become very sleek. And if you look at your mobile phone, which are very small, but they can actually do equal or even better job than many of these earlier days computer. That's because of this the circuit, miniaturized circuits and chips, which actually has been very small, as much as you can actually make it. So you can make the chips in nano dimension, that is what we call nano chip. So you can actually load more data into it, and they have become faster. Similarly, energy, you can nowadays see the solar cells a lot. They are basically made up of silicon. You are now getting to see some of the electric vehicle coming to the market. And then this is a flexible electronics where you can see that you can actually bend even the solar panel. So this is also medicine aspect. If you look at, I think last talk was excellent on this drug discovery. Nowadays, people are talking about nano formulations of the drug, and also the targeted delivery based on the nanoparticles. So I'm going to give you a flavor in next couple of slides before I actually get into my own research. So these are various aspects of it. You can see in fact, not only that, consumer goods like many of these creams, they also have nanomaterials to protect your skin. Now, in that regard, I must tell you that why a government is also nowadays giving a lot of thirst on these nanotechnology based product and nanosciences is the following reasons, because we feel that these are the major challenges for the mankind or for the country. That is, we all know the problems due to energy. We know the fossil fuels are limited, and we need to have a renewable source of energy, right? Then environment is another aspect which we need to protect. So can we really mitigate these challenges using nanotechnology? So these are interconnected, because more you burn fossil fuel, they also destroy the environment. So these are important from the renewable energy aspect. Security, then nowadays you need to, if you pass through airport, you always pass through the security, and this has to be an the more there are threat from the terror organizations, and their detection limit of the bomb. So that means you need to also develop some new and very ultra sensitive sensor to detect that. non-invasive method, in a non-invasive way. Similarly, food is another thing. Food and agriculture, high throughput agriculture, that is also important without compromising the fertility of the soil. Then again, disease control. So these are very important aspects, and I think nanotechnology is important. Now here I would like to also share with you, nanotechnology is not a different stream. It is actually all coming from this chemistry, physics, biology, whatever you specialize in. Nanotechnology comes as a branch later. So there is nothing nowadays. Many of the universities are coming with MSc nanotechnology and all, but you need to have some specialization. Something should be your USP. Let's say you are a chemist here. So you always look at the nanotechnology from a chemist point of view. But remember one thing, when you actually later on pursue your career in science, research and development, so it is not only that you should know only chemistry, you should also know a little bit of physics or biology. And that is the reason I actually wanted to incorporate few 10-15 slides of my research, just to give you a flavor that how, being a chemist, I come from a chemistry background, I now need to know a little bit of physics or biology. So that means what I tend to imply is that don't ignore your past subject. Even if you have a chemistry honors, you must take even your physics or biology, whatever past subject you have, very seriously. Okay, now first thing, as you know, energy. So energy problem mitigation, as you know, solar cells, they are very everybody, all of you know solar cells. They are from p or n type silicons, which are actually semiconductor. This much probably you know. And they actually, because of the presence of light, they actually sorry, but yeah, so band gap of these semiconductors are very important for the solar cell efficiencies. And if you can see, the silicon solar cells can have a maximum efficiency of something like 25%. And there are many of these different material which has come later on. And as you know, in order to make this, you need to know chemistry. And especially here, in this particular case, you need to be very good inorganic chemist, so that you can make this material and actually fabricate onto these solar cell devices. However, one thing is you have to accept that silicon is very cheap material, but many of the materials which are coming, that they are relatively expensive. So there is also research and development going in that direction to make cheaper, in comparison to also the efficiency parameter nanomaterial, which can increase the efficiency of the solar cells. So as of now, this is very important, because efficiency of the solar cell, if it is lab scale, it is 25%, it might come down to 10% only. And that is the reason you will see solar cell can only, if it is there in your college, you will see it can only run your fan or light, but it cannot take the load of a air conditioning machine or some heavy load equipment, because that requires more power generation. And for that to have, you need to have really a huge area of solar panels. But that much area, especially in state like West Bengal, we don't have that much area to put the solar panel. That means what you can do is that instead of so you can change your silicon solar panel, solar photovoltaic cells with some superior material, which is what can push your efficiency to something like more than 50%. So that should be the dream. And there is always ongoing research and development. So this is something which is a very interesting area if you are taking up in inorganic chemistry in future. So similarly, this is also energy. So you can also consider hydrogen as fuel, but there is also a problem. Hydrogen you cannot store because it's very flammable. So there is a problem. So you can make some network-based material and you can actually store hydrogen as fuel. And using this, you can see this train is running as hydrogen as fuel. This is in Germany. And there's also another thing nowadays people are doing is that you can actually consider water as the fuel. And you can actually have photocatalytic water splitting, and you break the water into water and break the water into hydrogen and oxygen. You can sequester the oxygen and separate the hydrogen and use that as a fuel. That's a very cheap source of energy. For that, you need to actually develop very nice photocatalyst. For that also, you need to have very good inorganic chemistry knowledge. You can take up. again, if you look at, this is a typical fuel cell battery. And nowadays, if you look at your mobile, to many of these, the laptop I'm giving lecture from, the battery concept is changing. They are all now graphene based battery. The concept of supercapacitor has actually come in, which you can have very high cycle life as compared to low cycle life of the traditional batteries. So these are things one can actually look up in future. There's also another thing, something called piezoelectricity. So if you actually provide some more motion, it can, like I'm raising my hand, it can actually generate energy. For that, you need to make material. Actually, I am also involved in this particular field. You can actually make some polymeric material that can actually sense this motion and it can generate energy. So you can already, there are some products which are in market. So you can go for jogging and put this particular piezoelectric device onto your arms, and as you jog, because of your muscle movement, it will actually create energy, and you can actually charge your mobile phone. So this is very, again, since we're talking about electric vehicle, you see later on, maybe 5-10 years down the line, as the government mandates that there will not be any diesel vehicle, all will move towards electric vehicle. Probably you are going to see something like this, where it is the electric vehicle charging station. But remember one thing, now when you go to a petrol pump and go and fill your petrol, that hardly takes 1 minute, and you give the money and then go away. But if you remember your simple mobile phone and you want to charge it, nowadays only it takes time, like it takes at least 2-3 hours to get fully charged. So if you're talking about an electric vehicle, that means you don't want that you go to the this electric vehicle charging station and wait there for something like 1 hour to charge your charge your vehicle and then pass. That's not realistic. Then they will not be successful. What you need is that you go there, zoom in, and within 30 seconds you want your electric vehicle to be fully charged. So for that to have, you need something called exponential charging capacity. So that is also another direction the field is moving towards, and that's where also this nanotechnology is helping a lot. All this catalyst or piezoelectric device I'm talking about, these are all coming due to nanomaterials. So another aspect, I think this was discussed in the last topic, that being an organic chemist, how you can actually make drugs, and how, we talked about, Dr. Das talked about pharmacokinetics, pharmacodynamics, all the different aspects which are very critical. Now, nanomaterials also can be used very interesting way to deliver this drug at a targeted site. Normally, if you take a drug, let's say orally you take a drug, that eventually goes through your stomach and it actually gets absorbed in your bloodstream, and it actually goes around in your bloodstream. And what happens, suppose if you have a tumor, something over here, it actually circulates everywhere in the body, right? And it gets accumulated in all the organs, unless and otherwise you have a targeted delivery. That means if I post a letter from Mohali to Bethune College, if I write everyone, let's say principal, Bethune College, and I write West Bengal, then it will go to West Bengal, but it will take a lot of time for the postman to find out, oh, Bethune College is in Calcutta, and let me do it. But if I put the full address, okay, principal, so-and-so, Bethune College, then Calcutta, West Bengal, then definitely it will be faster. This is the same thing happens with the drug delivery. So what I do is that targeted drug delivery, you can put it in nano material, and it can actually target it at a certain place, and you can actually put certain markers which can actually differentiate between healthy cells or cancer, like tumor cells, and it can actually deliver. Like this is a typical intravenous drug, let's say, just for pictorial representation. It is actually floating all throughout your body. But if you actually make the nanomaterial and as a magnetic nanomaterial, which actually carries the drug, and if you apply a magnetic field, you can actually concentrate all the drug into this particular tumor, and this drug may not go to other organs in the body, which actually would cause you the side effect. So this is why nowadays research is going towards this nano formulation and targeted drug delivery. This is another very important aspect. One can, being a chemist, you can actually make those molecules. Not only you can make drug, which Dr. Das discussed, you can also make, you can also design, make by organic chemistry. So this is where organic chemists should go. You can actually make very interesting what you call is that vector or material which can carry the drugs. Okay, so now let's come and discuss about the title and insight into Nanoworld. So now, by now, you already know what is nano. That means I spoke about mostly about the dimension over here, and then I gave you some of the interesting application so that you actually can relate very cutting edge application where we are going to go in future in 10 years down the line, so that you can correlate where you are as a chemist and which pathway you should take to go towards that. So these are just inspirational, some of the slides I showed, but now I'm going to show you some of the actual figure, how it evolved. So if you look at in Nanoworld, if you look at an ant, which is a 5 millimeter size, but you see this is a human hair, which is something like 10 or 50 micrometer. And actually through your naked eye, you can only see one single hair, because below that dimension, it is very difficult for you with your naked eye to see that particular dimension. That is why if you go further down, something like 2 to 5 micrometer, that is actually a red blood cell. That is actually that you don't see through your naked eye. You rather you need a optical microscopy, right? If you look at DNA, so if you look at DNA, this is double helical DNA. That is actually the real nanomaterial, because that typically that comes in the range of this 1-2 nanometer or something like that. That's actually typical nanometer DNA. How can you see DNA? So that's a question I would like to keep you for till the next slide. Similarly, looking at these which are present in natural world, man always wanted to make something which is which we can mimic nature. And similarly, we also have made carbon nanotube, which are also very similar to the DNA dimension. Now, carbon nanotube is also very important, and I think today, I mean, I also go and tell the school students that look, if I ask you different electrons of carbon, that means earlier days people used to give answer that it's only the graphite and the diamond. But I think in today's world, you should know that there is also fullerene that is important. And then there is something called these carbon nanotubes, which are also very important, these are all carbons. And then there is also something like if you take a scotch tape and stick it on the graphite surface and pull it up at once, you will see one layer of graphite will come out, that is called graphene. So these are some of these interesting nanomaterials which has really very huge implications. So if you have ever flown Air India, that is you know about Air Dreamliner, right? That is Boeing 787. Now, you know this, normally now I want to give you one very interesting perspective of it. Normally what you think is the very heavy material that we by default think iron or maybe steel, right? That's an alloy that we consider is very heavy, but that's very robust. But on the other hand, if you talk about the lightest material or metal, that is actually aluminium. And that is why you will see that aluminium is being made for all these rocket or let's say in flight, because this is very light and you actually have the buoyancy effect so that they can actually replace equal amount of air and they can actually fly easily. But remember one thing, when we send rocket from here, let's say Sriharikota, which is like 40 degrees Celsius, or to the high up there in the space, you see there is a huge temperature change which happens. And as you know from your class 5 or 6 science studies, you know that there is a gap between the railway track, because what happens in summertime, they expand. So there is this expansion contraction happens. The carbon nanotube is one such material which has very low such expansion or compression coefficient. So with temperature, it does not really change its dimension so much, just like our aluminium would do. And that is why if you send such material based rockets up there, there is a high chance that it might be failure because of this temperature gradient, it may actually burst. So nowadays, a lot of these carbon nanotube based polymer composites is and also there are some other superior material they are being used in by ISRO or other space agencies, so that you don't have that expansion contraction and you have mission always successful. So these are some things which are very interesting things. But now, as I was saying, you can, how do you see it? Like you are seeing me, right? How do you see me? Because the light is coming and falling on me, and then it is going into your eyes through this particular zoom camera, and then you can see me. But remember one thing, what is the visible light wavelength? 400 to 700 nanometer. In order to see me, that means I am the object. If you have to see me, the wavelength of the light has to be lower than me. That's obvious. That's a basic thumb rule. Okay, now what happens is that when you talk about a red blood cell, that means it is 2 micrometer, something like 2000 nanometer. Now 2000 nanometers, still you can see through the visible light, because visible light wavelength is still lower than the 2000 nanometer, the dimension of a red blood cell. So, but in that case, you cannot see through your eyes. That is where your limitation is. So what you do is that you put an objective lens. So you make your eyes actually empowered with this objective, and you can see through optical microscope. I'm pretty sure that all of you have actually done some experiment or others through an optical microscope, like cutting the cross section of your onion, right, or something in +12. But now, as I already talked about, if you have to see nanomaterials, real nanomaterial like DNA, 2 nanometer, definitely you cannot see through visible light. Why? Because visible light has higher wavelength than the nanometer dimension. For that, you need to have electron microscope, where electrons are used as the light source. And I don't need to really explain to you the wave particle duality if you are studying quantum chemistry. So you know it very well that it can be particle, or it can also have these wave-like features. That is what is very important. And when this electron microscopy came into the nature, people started realizing these nanomaterials in a different level. Just like I discussed about the story of gold nanoparticle. See, we all know that the leaves are green. Then you have these carrots or anthocyanin. These are different, colorful. The world is very colorful, and we know that there are some pigments which are responsible for it. So what happens? This pigment, it actually absorbs the color, and the complementary color it cannot absorb. So it actually get to see that particular color, right? It based on the absorption and emission of that particular concept. But then the scientists could not actually explain why the butterfly, why butterfly has so many different color on this on its wings. And if you actually extract butterfly wings and extract the pigment, they don't find too many different kinds of pigment. So they were really puzzled. Why is it so? You don't have that many pigment to account for so many different color. Why is it so? But when the electron microscopy came, they started looking closely through the microscope using the wing, and you can see that they can have this nice thin fin over here. And if you look at cross section way, you see there are these air pockets, and these are all periodic in nature. Now what happens, all of you also know from your class 7 physics, that if you take a stick and dip it in a bucket full of water, the stick look apparently bent. Why is it so? It is due to the different refractive index of water and air, and that is why you get to see something called refraction, or in Bengali all of you say that protisharon, right? So what happens here, that particular refraction keeps on happening periodically many times. Okay, and that actually gives rise to some new incident called reflectance, something like reflectance. You can read this paper, which is rather recent. Now, seeing this, scientists thought this is very interesting. So we don't need to have different pigment. We don't need to really do a lot of tremendous organic synthesis to make different pigment, color variation, so that you can maintain the Woodward-Hoffman rule. I think you are studying that all, so that you can have this double bond, right? You change the electronic absorption and all this, right? So rather, what you can do is that you can actually make nanomaterial in a patterned surface, and you can generate different kind of color, just like the butterfly do. That's what nature does. So that actually gave very interesting insight. And then that's why a lot of different industry boomed after that on photochromism and such industries. Now, another interesting aspect on this pattern nanosurface. So all of you have seen these lotus leaves, and also you know something called taro root, pochupata. So you will see that one particular side of these leaves, they have a different kind of texture, and if you put a drop of water, it goes into a spherical shape, and it comes due to surface tension. And if you look at now through electron microscopy, you see there are very nicely patterned this kind of beads. If you ask someone, you said, oh, there is some layer of wax there. But if you actually look at more scientifically, in today's understanding is that you have this pattern surface, and because of that, the lotus on the lotus leaf, the water droplet, they minimize this contact, and this can be also defined by something called contact angle. And for the contact angle, which is something like this, very high contact angle, you call this surface as super hydrophobic surface. That means if you have super hydrophobic surface, so your water molecule cannot really stay there, and it actually, so your dirt also will come there, but if you flow water, then what will happen? Just dirt actually has fallen over here, and you just, with a pipe you put water molecule will be round in shape, and this will just take the dirt away. So this is very interesting, and we learned from it that, okay, we can make interesting nanostructure so that you can make. So nowadays you will see PPE kits. Okay, so they are also coming up with the hydrophobic surface. Okay, now another interesting thing. So learning that, you can also make very interesting textile that can be used for nanotechnology, something called nano finishing. So you can make a textile where you can have this super hydrophobic like water droplets. So there are already marketed products into it. It has already come, and these are called self-cleaning textiles. Okay, now look at some of the interesting example. Like you have beetle or your fly. If you look at their toe and their nanostructure, you see as you go from beetle to the gecko, you will see there are a lot of these fine nanostructure present where they actually trap the air, and that is the reason they can actually stick to the wall. So just because they have difference in nanostructure inside this toe, they can actually stick to the wall and actually move. And based on that, scientists, they get the idea of making this kind of sticky note, or the tape, where you can have this polymer fibrils if you can make, and if you can make it in the if you can patterned way, you can actually make such kind of very interesting sticky note. So these are the inspiration we get learning from the nature. Now, what do we do? Yes, we do also. So this is where I'm going to talk to you for next 15 minutes about our research, what we do to mimic nature. And I take my inspiration as a chemist from this famous lecture series by Richard Feynman in 1959. That's more than 60 years ago. He said that there is plenty of room at the bottom. That is, before even the nano science term was coined, he said so. That means he actually knew about it. And see, if you have to make a nanomaterial, there are two ways to make it. One is top-down approach or bottom-up approach. So what physicists can try to do is that you can take a material which is micrometer, and you keep breaking it. And as you keep breaking it, you actually get into the nanomaterial regime. But as a chemist, what we do, as we know, all of us do actually some reaction, covalent synthesis, right? So those molecules are something like in angstrom level, right? Now, we all know that 1 nanometer equals to 10 angstrom, right? Now, if you can take 10 such molecule and self-assemble them using something called supramolecular chemistry, which is not guided by covalent interaction, it is rather through non-covalent interaction. So you can actually make some self-assembly. So this is called self-assembly because you are taking few molecules and they are through non-covalent interaction like hydrogen bonding, host-guest interaction, pi-pi interaction. I think all of you would be knowing in your undergraduate all these interactions. So this is what we do as a chemist. We always prefer taking molecule, self-assemble them. And this interest actually grew when the university I came out from, where Bernard Feringa, he got the Nobel Prize in 2016, just for making a small nano car. So this is basically an organic molecule where if you shine light, just because you have a cis-trans isomerization, this particular molecule, they actually start moving. And I know the struggle he had for 10 years, because all these different wheels were initially moving in different directions. And that is why, as a whole, these molecules were not moving. So they had to apply their chemistry knowledge, and eventually in 2011, they cracked the case. And that is why after 5 years, he got the Nobel Prize. Incidentally, when he published this particular paper, I was in Groningen. So these two gentlemen, they also made very interesting something called nanolift, where it is a rotaxane-like molecule, and it is just like a shuttle. You can visualize that this man takes lift here, this goes up under some external stimuli, and actually you can work out. So this actually makes us inspired to design material in a bottom-up approach. Okay, now coming to some of the research, more inspiration I get looking at the nature. Now, if you look at, there is something called self-healing. I think healing, all of you know, right, this term. Now, automatically healed material is the one which I am interested in. And this is what you actually get to see in the tree. Let's say if someone cut a tree, then you see this actually has its own self mechanism, that it actually secretes some kind of latex-like material or some other kind of material where it can actually protect against some microbes, bacteria. So it has this self-healing mechanism. Similarly, this chameleon that actually changes its color because of reversible muscle control. Now, this is what exactly I'm talking. So if you have cut the stem of the tree, what happens? It has this inherent mechanism to heal this. Similarly, in our body is also made up of, you see, if you have a small cut somewhere, it automatically heals. But if you have a bigger cut, of course, then you need medication. You also need bandage, all sorts of dressing just to heal that, something called wound healing, right? Just to facilitate this healing process. So now, if you are thinking of material, smart material, where we can actually implement such understanding what we have, so people, you see, started making very smart material where you can have something called extrinsic healing material. So what you do is that you make the material and then you make small microcapsule, and inside the microcapsule you put some healing agent. So as soon as you cut it, as you cut it, what will happen? The microcapsule will also get ruptured, and the healing agent will get released, and it will actually heal the surface, just like tree does, right? Latex is the healing agent in their case. In this case, our healing agent will be some chemical which will be inside this microcapsule, right? So this is something called extrinsic. Okay, but now problem is that you can, once you break this microcapsule, only once this will come out, and that's it. So only you can heal it once. But in our body can heal many times, right? If you have to mimic it perfectly, then you need to have multiple healing. And that is where you need to have these different stimuli responsive chemical approaches, which is intrinsic in nature. So you need to use supramolecular chemistry, which will have hydrogen bonding, pi-pi stacking, guest host chemistry, metal ligand coordination. So you can see organic chemistry, inorganic chemistry, physical chemistry, all the things you can actually cover if you are working on such material, and you can actually have multiple healing. So now another interesting thing I would like to show you here. Now this is where polymeric material comes in. So you make polymers and you cut the polymer, and you want the polymer to heal its own. That's why we call them self-healing polymer. So this is an example of such self-healing polymer. You see, they make the polymer, you cut it, and then it can actually automatically heal. So in normal polymer, you may have, particularly for thermoplastic polymer, you will see there will be two different regime. One is very flexible part, another one is little rigid part. Okay. Now, when we see protein folding, now I'm just giving some idea. Protein is, as you know, it is a polypeptide chain, and when it folds, it actually gets some definite shape. You have seen in the previous slide, and it has some definite function. Now, when it folds, it actually encompasses some of this supramolecular interaction into it. Now, wherever there is a cross-linking because of this physical linking, there will be some rigidity which will come. Otherwise, the as such protein molecule may be flexible, but those are the cross-linking points where you may have some rigidity imparted, right? So based on that, we thought, okay, now can we have a polymer which will, where we'll put some functionality and we'll make the polymer chain collapse just like in protein folding it happens, and but wherever the cross-linking happens, there will be some rigidity which will be imparted, which will give something called flexo-rigid polymer network. So why, being an organic chemist, what I can do, I can synthesize this polymer compounds, and the functionality here is something called coumarin moiety. Now, I think I should also tell you coumarin is very biocompatible. It is also found in lavender. The interesting thing of coumarin is that coumarin has this double bond, which can actually, if you have studied pericyclic reactions, it can actually have this cycloaddition reaction over here, and it can form this cycloaddition product, and this is reversible. If you have different wavelength light, you can tune it reversibly. And based on that, so these are the different polymer we made it. As I was saying, that we can have this polymer synthesis, and this we can control through the light. I'm not going to go much in nitty-gritty of my results because that is immaterial at this point. I will talk about the implications. And we have shown that if you can cross-link the polymer or uncross-link the polymer, based on that, you can actually have the time of the self-healing, you can modulate. So you can see that it is rather flexible polymer. You see that in 10 minutes, it can self-heal. But if you make the polymer much more rigid, even after 10 hours of irradiation also, it does not really heal. Okay. So we have shown this, that we can actually have automatically self-healing through autonomic self-healing or non-autonomic self-healing, where you need to have a particular photo stimuli required, which will, as soon as you irradiate, it will start healing it. So this is an interesting thing which can be applied. See why I'm saying this, because you can have these nice photovoltaic cells, right, for solar panels. Now what happens, because of the pollution and all, it may have an acid rain, right? The acid rain may corrode the surface of the photovoltaic cells. Over the year, what may happen? Over the year, what will happen, that this will corrode the solar cells, and it will degrade solar cell efficiency. So it will not last long. So if we can make this coating on the solar cells, very thin coating, what will happen? It will protect it against this acid rain, or let's say some bird comes and fly on it. Because of the toe of the bird, it gets damaged. But because of this healing nature, it will automatically heal, and it will actually make the solar cell, let's say in this particular case, last long. We can also think of something like scratch-proof surface on the wall, where it can automatically heal, because paint, actually you can see that they sometimes crack. It can automatically do, then your house does not look good. So you can make all these different things. If you talk about, again, this is what you control is here, the nano dimensional manipulation you do, and you can actually control such properties. Now, another thing we are working now, I think all of you have seen this kind of let's say earphone, and you have seen that after some time this earphone you pull or something happens and it does not work, right? And that happens because these are all polymeric material, whatever you see, or any device you take, and any of this cable you take, they are all polymeric material. Okay, now that is basically, polymeric material is nothing but organic molecules which are monomers which you polymerize, right? Now what happens, sometime if you pull or there is a high voltage, then there will be some crack in the polymer, and because of that crack creation, you can see something called, this phenomena is called electrical treeing effect. That is where you see that after some days this particular earphone will not work or something happens. But now if you are thinking as a future, developed country like in India, maybe all the cable will be underground, maybe 20 years down the line. But if something happens in the bottom, it will be very difficult to take out and repair and replace, particularly in city like Kolkata, right? So what you need is the cable which can self-fill it automatically. So this is the implication. So we are now making some of the magnetic nanoparticle. One of the interesting aspect of magnetic nanoparticle is that when you apply high voltage around it, high magnetic field, electromagnetic field, this particular magnetic material generate heat, and this heat may actually melt this polymer above their glass transition temperature, and it can actually make it again healed. So these are very interesting applications which actually can come. And again, this looks much more like physics aspect, but again, I'm saying, being a chemist, I should be able to make this material so that I can talk to my colleague, understand the physical aspect and discuss. So this is what is needed in India so that you can pursue research in such dimension. Now, this is interesting. Okay, from the point of view of drug, as the previous lecture talked about this drug delivery. So nanoparticle as drug carrier is also very important. Now, what happens is that, normally the drug actually circulate in your body through the blood. Now, if it has to go to brain or if it has to go to eyes, they have to cross something called blood-brain barrier or something called blood-retinal barrier. Now, this will actually excrete and keep your brain or eyes, safe, normally, the normal body. But if you want your drug to go there through some nanoparticle, then what happens is if your nanoparticle is very big, they will not allow you to go inside the eyes or brain. And that is why it is a challenge to deliver the drug in such brain or retina. So that is why we are now working on nanomaterial, again, organic nanomaterials, which are very small in dimension, something like less than 30 nanometer or so, and this can be thermoresponsive. So as and when, suppose you actually take some antipyretic drug, that means if you have got a fever, it will, as soon as it senses that okay, you are getting a fever, it will actually release the drug. Otherwise, it will not release it. So you will not have much side effect. Such is the thing, such is the idea. And as you can see, this is again TEM images, and we can have very small nanoparticle we can create. And I'm not going much into the details of it. So this is the idea towards it, just to show you that you can actually have very interesting this kind of polymer as the drug delivery vehicle. Okay, so we also tried to look at it, how is the drug release, how fast the drug can release. You can see that based on different kind of nanoparticle you take, you can either release it very fast within 5 hours, or it takes really 24 hours. So you don't need to have different doses. You can already control in your body the doses. So now I think I don't have much time. And another interesting aspect, being a chemist, you can look at, this is very fundamental. This has nothing to do with nanomaterials, but I thought this is also very important for us to know how we got created and how to model them, how we can actually, model how life came into earth. And all of us know that, we had the primordial soup, where you have this precursor of many of these biological compound like amino acids, nucleoside precursor, which is DNA, lipid precursor. They were there, something happened, then they start making some molecule, and those molecule, what you make, they actually start to self-assemble. And not only self-assembly, you actually have self-replication to make this kind of compartmentalized self-assembled lipid vesicles. That is where eventually your protein or your RNA, DNA, they actually get inside, and they eventually form something like protocell. And as you know, if you take many of these different cells, they actually make tissue. So this is actually, if you can model it with your chemical compound, then you can actually understand the origin of life principle. So you need to know good self-assembly. You need to know self-replication. That is very important, because self-replication needs to be exponential, because it has to grow very fast. Otherwise, life will never be able to sustain itself. It has to be exponential. So these are different aspects we are looking at in our lab. I don't want to go much in details, even though I have a couple of slides. But for that to know, now again, coming back to how to mimic nature, you see, this is the typical honeycomb, which is very, very hexagonal structure, you see, or the water crystal. This is automatically done by nature. Nature is a perfect engineer. But when we do this self-assembly in our lab, try to make exactly like this, we faced problem. And last 10 years, people started doing it. And for that, what we are changing, I think that is also very important aspect. This is again, physical chemistry point of view. I need to know that. So normally, if you are making, you are doing some reaction, you have always kinetically controlled product, thermodynamically controlled product, right? That's a covalent chemistry. But when you talk about self-assembly, that is non-covalent chemistry. And you can have this particular product, which is thermodynamic product and thermodynamically controlled product. And if there is some higher energy, if it is there, that is dictated by the thermal energy. That is due to the Boltzmann constant, right? You must have studied already. But this is called self-assembly you are doing in thermodynamic equilibrium. And this is what only in a lab as a scientist we can manage. But what nature does is that nature actually can have very interesting things, kinetic self-assembly. So here nature can have even population at the higher energy state, keeping the lower energy state vacated. This nature can do. So in order to mimic such a precise nature material, we need to also know how to trick with this kinetically controlled self-assembly. So this is what in our lab we do. And we take inspiration from amyloid like protein. I think previous speaker discussed. We pick out some particular amino acids out of it. We synthesize some particular peptide molecule, peptide fragment. These are all amino acid, typical organic structure. And through atomic force microscopy, you can see different kind of, these are nanoparticle. You increase temperature, it actually forms fiber. It goes into twisted bundle. So you can actually control the self-assembly pattern. So these are things we do. We can also control, again, using this Kumarin photodimerization, you can also make two-dimensional material, one-dimensional material. This is again from the organic chemist point of view, and you can actually do a lot of this interesting nanomaterial, because making very precise material is something as a scientist one would like to know, and this is where we are going. And this is a very recent paper, and the implication is the following. You must have also studied Louis Pasteur. Louis Pasteur, what he did is that he actually separated these different stereoisomer of sodium ammonium tartarate, right? So left-handed and right-handed, looking at the optical microscopy. So we thought, can we really have now L type of peptide and D type of peptide? And if you throw them together, can you really do something called self-sorting? What is self-sorting? Means, in Bethune College, if you have people coming from Medinipur, people coming from Malda, and people coming from Kolkata, let's say first day they come, they are all intermingled. After 7 days, you see all the Medinapurian people, they are going together, all the Kolkata students are going together, and all the Malda students, North Bengal students are going together because of the region, right? So here region actually dictates that, okay, they self-sort among themselves. Similarly, if you make now material, something L-based peptide or D-based peptide like this, and if you throw them in solution, if they can segregate automatically based on their molecular recognition, then it is something like, what Louis Pasteur did 200 years ago. And that's what actually we showed through super resolution microscopy in nanometer scale. These are all in nanometer scale. You can see we can have L peptide, which are green fluorescent marked, D peptide, which is red fluorescent marked. These are the typical thing. We have to do all the organic synthesis. And you can actually see all these different fibers very nicely. This is something which has got accepted recently. And well, now last two slides, I would like to say that based on this peptide material, you can also make something called hydrogel. Now, this is very interesting. Even in ISS, you will find a lot of people who are working. Now, hydrogel means you take your chemical and put it in water. What it will do is that it will actually self-assemble and make some kind of this three-dimensional fiber, and it will actually prevent the water from flowing. Okay. Now, hydrogel is very important because you can make very nice of this patch, and this because this has mostly water. So if you have a burn injury, you can actually put over your burn injury and it can heal, and you can put your drug. So it will be faster than earlier days people used to do this bandage. And diaper, it can actually soak up this water because it has those hydrogel pipe from the cellulose fiber. Similarly, nowadays 3-dimensional printing is coming, 3D printing, what you call, and you can actually, using this stem cell research, you can also make this artificial organ, something called tissue engineering. So even though I have 2-3 slides, I think I'm going to finish it. So this is application of hydrogel. You can see that this, you can with the hydrogel, you can see that it can heal the wound much better way than the normal dressing you can do after a couple of days. So this really does a fabulous job. I work on tissue engineering from the peptide based gel. So basically what you do is that you take your stem cells, put it in this hydrogel scaffold, put it in your body, and then grow the cells. And this is specifically, I'm working on bone cells. So this I'm not going to discuss much in details. And another last slide I want to finish up with is that if you have to really mimic artificial material, like you look at these heart muscles, very interesting, because it is the heart muscle, they have something called non-linear elastic behavior. Because as you know, heart has this systole and diastole, right? In one case, it actually compress, and another case is actually expand. Now your material, which actually makes these cardiac muscles, which are nothing but these, protein-based collagen material or actin filaments, so they need to show these something called non-linear elastic behavior. That means when you apply force, they will behave non-linearly. That is very important, this non-linear signature. And if that is not there, what will happen? Your heart will rupture and you will die. And that is why if you are making hydrogels, we don't see this synthetic hydrogel has this non-linear elastic behavior. They are linear in nature. That is why you cannot do this effectively tissue engineering. So our group, we are working on this non-linear elastic behavior based material using some physical concept. This is again a physics paper we are following, and we actually design material. I'm not going to show all the details. So we are working towards that. And I hope I just showed in what I showed about the self-filling. I showed about making different nanomaterial, how to make these orthogonal self-sorting. Biomineralization is last 10-15 minutes. And all the work we are doing with these very motivated students. And I also thank my professor, my director, Professor Amitav Patra, for encouraging me to give this lecture to Bethune College, so that all of you actually get a feel for the nanoscience. And I thank you all for your attention, and all the scholarship slide. I thank to Dr. Das, who has already showed all the details. So I removed all of them. And in future, if you want to really visit our, you can make an educational trip to our institute. We have a very special outreach cell. I am the chairman of that. So we can make your trip over here, stay over here. You can visit the lab, learn something, and different aspect we can discuss. So you're most welcome to visit at any point of time. Thank you. Thank you for your kind attention.
Thank you, sir. Thank you very much for introducing this topic of nanoscience to our students, telling them so many different varied kinds of applications, as well as telling them what can be the career opportunities in nanoscience for them. There are many questions from your interesting lecture, both from YouTube link and from the Zoom link. I don't think we'll be able to take all the questions because we are running already short of time, but I will request you, sir, to just answer a few questions. Shall I proceed?
Sure.
One question is here. What are the most common approaches?
Name the person, that will be also good.
Yes. She is Debaruti Dash. Okay. She is Debaruti Dash, and she is asking, what are the most common approaches for synthesizing nanoparticles with different shapes? What kind of parameter control is required if we go for normal gel combustion synthesis or solid state reaction route?
Yeah, I think this is very technical question. I think this question, yes, there are a number of parameters which are there. You normally, you can always take the capping agent ratio is important. I'm just giving a general answer, not to very exactly. These are things we need to really, when you are doing some nanoparticle synthesis based on, end cap ligand, you need to optimize the concentration of the ligand. Then the temperature is important. Then, if you want to control the shape of the particle, and after making the nucleation, the growth, there is something called Oswald ripening. You can actually, control the growth. There are a number of parameters which are there. I think this, if you write it separately, email, this can be, we can cite some of the papers, and some of that very interesting paper, you can look at it. Again, coming back to nanomaterial, there are diverse type of nanomaterial. Very, very, very hardcore inorganic materials are there. Some of the parameters would be completely different. Some organic nanomaterial, which I discussed here in my topic, yes, I can change, let's say, the polymer chain length, the functionality control, grafting. So different, these are all again going into the subdomain, and you have different kind of parameters. So yeah, it's a very general question. If I cannot answer it in this particular, if you have a very specific question, you always can write, then I can help you out in that. Yeah.
Okay, sir. So we will take just only one question from this YouTube link. What are the most common approaches for synthesizing nanoparticles with different? Okay, so this was the question which was already asked. Are all nanomaterials green in nature?
No.
Are all nanomaterials green in nature?
No, not really. I mean, there are materials which can be also toxic, right? So no, I don't think it cannot be generalized that way.
Okay, sir, thank you. And for all other participants who have a lot of
I
questions.
think I would like to also say one more important thing. This I think I must complete. Yes, one thing is that yes, nanomaterials are green or not. No, that is one thing. But another thing, as we say, nowadays we are making a lot of this nanomaterial, doing drug delivery. But I think, as the previous speaker also talked about, the regulatory body. So now in the nanomaterial research also, particularly for the health application, we are also looking at the regulatory body clearance, because what happens, nanomaterials are so small that they cannot be also trapped by any filter. They are going out from the body, let's say, getting excreted from the body. They're going into the soil, right? And that might, if there is a toxic effect, of course, they might also create it, pose hazardous challenge for the environment. So this is something also we must look at it. And already the nanotoxicology groups are looking at this particular aspect. Yeah.
Thank you, sir. Thank you for the answer. And I think all other participants, I think you can put your query through email to sir. And sir will ask your give your answers to your queries. Now, I will request Dr. Amita Kaur to deliver the vote of thanks and conclude the webinar. And for participants, the link for the feedback is already given in the chat box. Please fill them, and you will get your certificates within 7 days. Amita, please. Amita, please unmute. Amita, please unmute.
Unmute. Thank you, Paramita. I hope you all can hear me now. Good evening to everybody. And we've just had the experience of enjoying and listening to a wonderful webinar where we traversed from the fascinating world of drug chemistry into the fascinating world of nanoscience. And like me, many would have liked this webinar to continue, but all good things have to end at some times. And as we are running very short of time, I would like to say goodbye. But before saying goodbye, I would like to thank all of you all who have participated in this webinar. I would like to thank our principal, Professor Krishna Roy. It is because of her inspiration and support that we could organize this seminar. My heartfelt and sincere thanks, of course, to Dr. Parthasarathi Das and Dr. Ashish Pal for giving us their valuable time and sharing with us their thoughts and experience for the future prospects in the world of pharmacology and nanoscience. I'm sure the students will benefit from it and will be motivated to pursue their careers in the field of pharmacology and nanosciences. Sir, thank you to both of you all, and we would like to meet both of you in person in near future when the situation improves. I would like to extend my thanks to the Dr. Kamal Kanti Shom, our IQAC coordinator, and also to all the members of the IQAC team of Bethune College for their support in organizing this seminar. I would convey my sincere thanks to, of course, all the participants, the teachers, and my dear students and all others. And it is because of your participation that this webinar has become a success. Special thanks to Dr. Srirupa Banerjee for a very nice welcome address, to Dr. Gautam Bandyopadhyay for his very well delivered introductory speech, Dr. Paramita Majumdar for anchoring this session, and Dr. Bimal Kumar Dirghangi for co-hosting this session. I would also thank my departmental colleagues, Srimati Indrani Sinha Roy, Dr. Rita Sengupta, Dr. Sulakhana Kormokar, and Dr. Shukriti Pal. Though they have not come out in the forefront and are behind the scene, but they have put their all-out effort to make this webinar a success. And finally, I would like to thank Sri Akash Mondol of Braindrops for his technical support. It is because of him that we could have such a flawless transmission. Thank you all again. And before I conclude and say goodbye, I would like to remind you all again, please fill up the feedback form and of course submit it, and you will get your e-certificates within 7 to 10 days. The feedback link is available on the chat box and it will be there for a few more minutes. Thank you all again and goodbye. Paramita.
Yes, Amitadi. Thank you. Thank you for the vote of thanks. And I think Amitadi has very nicely concluded the webinar. It was a wonderful webinar. Thank you all. Thank you all for participating. Thank you, the speakers and all my departmental colleagues. Thank you.
Thank you. So we are signing off.
Yes.